Hey guys, and welcome back to the lecture series. Today we're going to be talking about DNA biology with a focus on gene expression. As always, I've provided you some learning objectives to help you track through the lecture. And if you're taking my class, you should have access to my lecture handout in both Word and PDF format. You can have it up on a computer and you can type your notes, or you can print it out and write your notes out as we go along. But those lecture handouts really help to track with the lecture. So, without further ado, what we're going over today is we're talking about gene expression. But in order to understand gene expression, we're going to have to lead up to it. We're going to build up chronologically. So first we're going to talk about DNA, and we're going to focus on DNA replication. Then we're going to talk about gene expression with a focus on transcription and translation, making sure to distinguish this process from this process, right? And then finally, we're going to talk about mutation. We're going to talk about types of mutation, genetic disease, and where you see the frontiers or the major changes taking place in medicine, particularly molecular medicine. So, in order to understand DNA replication, it's important to understand a little bit about life cycles. So we're all kind of familiar with a human life cycle, or the life cycle of many animals, specifically mammals or reptiles or birds or whatever it is. And at some point in the life of a human, they consist of two independent cells. There's a sperm cell and an egg cell. Those cells fuse to form a zygote. That zygote undergoes many rounds of division called mitosis to form a baby. Babies grow. Those cells divide more and more to form children, grow into adolescents, grow into adults. These adults go out and serve some kind of function in society, right? Now, these adults are capable of producing sperm and egg, and the cycle begins again, but eventually these adults will die. So that's a human life cycle, a very brief description of a human life cycle. And just like humans have life cycles, individual cells have life cycles. We refer to the life cycle of an individual cell as being the cell cycle, right? And it transitions from one phase to another. So when we look at the cell cycle, it's broken into two major phases, two major times. So when you see this purple indicator right here within the cell cycle, the life cycle of a cell, you see that it's called interphase. Most cells spend the overwhelming majority of their time in interphase. Interphase is the time when a cell is just being a cell, when it's out there serving some kind of function, like a liver cell is, metabolizing toxins and producing proteins. That's during interphase. That's when a cell is just being a cell, right? And specifically GO, but you're going to talk about that we're going to talk about that when we get into mitosis. But interphase is when a cell is being a cell. So if you look over here at these epithelial cells, this cell right here, this was pulled from the lining of the cheek, and these cells right here, they're just being cells, right? They're lining the cheek, they're forming that tissue, they're producing proteins that metabolize sugar or structural proteins, and they're serving that kind of protective role. Now, eventually cells get old and worn out, or they need to replace cells that sloughed off. We lose cells all the time. We're constantly having to repair or maintain tissues. So cells need to undergo division. They need to replicate themselves. That division is called mitosis. So within the cell cycle, cells are essentially transitioning between interphase, where a cell is just being a cell, and mitosis, where that cell is actively dividing to produce two genetically identical daughter cells. And mitosis occurs for a constellation of reasons: growth, development, wound healing, maintenance of tissues. I mean, there's all sorts of reasons. Our cells are undergoing mitosis all the time. So in general, interphase is when a cell is just being a cell, and that constitutes the overwhelming majority of the cell cycle. And then mitosis is when a cell is actively dividing to produce two identical daughter cells. So you see this cell here transition from being in interphase to being in mitosis, and actually you can see the cellular division take place. So one cell forms two genetically identical daughter cells. Now, while a cell is just being a cell, it has to prepare for mitosis. And one of the things that happens during interphase when a cell is preparing for mitosis is it replicates its DNA, because if you're going to produce two genetically identical cells, you're going to have to double the amount of DNA so you can divvy out that DNA to each of the daughter cells that are produced from the process of mitosis. This is a special subphase within interphase called the S phase. The S stands for synthesis. And that's when DNA replication is occurring in the cell in preparation for mitosis. So it's not mitosis, it's a preparatory step saying, hey, we're eventually going to divide and we need twice the amount of DNA in order to make sure that both cells get an equal amount of DNA. So when you think about DNA replication, where does it take place? DNA replication takes place in the cell's nucleus. So here we have the nucleus of a cell, and around it we have the entire cell. This is just that central organelle called the nucleus. So where does DNA replication take place? In the nucleus. Why does DNA replication take place? It takes place in preparation for cell division. If you're going to produce two cells, each one is going to have the same amount of DNA, right? The same genes. So you're going to have to copy that stuff and divvy it out. Now, when you think about DNA in a cell, DNA doesn't just exist by itself in the nucleus of the cell. When we say the nucleus stores and processes hereditary information, DNA exists within the nucleus of a cell, but DNA is a really, really long molecule. If you were to stretch out all the DNA from all the cells in your body and you were just to lay it end to end with the 200 trillion cells, it would extend from here to the sun and back like 7 times. DNA is a ridiculously long molecule. And in the nucleus of every human cell, adult human cell or somatic human cell, you have 46 long strands of DNA, really, really long strands of DNA. Right? 23 come from mom, 23 come from dad. So how do we get those long strands of DNA to fit in this confined space of the nucleus? Well, we take that DNA and we wrap it around these proteins called histone proteins. So if I was to draw an analogy here, imagine a histone protein being like a toilet paper roll, and imagine DNA being kind of like the toilet paper. Now, in order to condense your toilet paper, you don't just have it floating around in your bathroom. Some of you might, right? But you don't generally just have it floating around in your bathroom. You take that toilet paper and it's wrapped around a toilet paper roll, and that helps you to condense it. DNA, which is kind of like the toilet paper in this analogy, wraps around the histone proteins, which are kind of like the toilet paper roll, and it allows DNA to condense. So when you have DNA wrapped around histone proteins, it actually forms a substance called chromatin. So DNA wrapped around histone proteins forms a substance called chromatin, and it's actually chromatin that exists in the cell's nucleus. Then if you need to use a region of DNA, like a gene of DNA, you can unravel it and you can specifically use that. So DNA wraps around histone proteins to form chromatin. When the cell is getting ready to divide, that chromatin condenses. And when that chromatin condenses, it forms a structure called a chromosome. So condensed chromatin is a chromosome. We have 46 long strands of DNA wrapped around histone proteins. And when the cell is getting ready to divide, not only does DNA duplicate itself, but it condenses. And when it condenses, we can see it, and it forms this structure called a chromosome, right? So condensed DNA forms that chromosome. If we have 46 long strands of DNA, we have 46 chromosomes, 23 from mom, 23 from dad. Chromosomes pair up according to corresponding genes or variations of corresponding genes. So here we're looking at what's called a homologous chromosome pair. Here's one chromosome from mom and here's another chromosome from dad. So you got a bunch of DNA wrapped around histone proteins forming chromatin in the nucleus. Now, when a cell is getting ready to divide, DNA replicates itself, meaning you copy all your DNA. That takes place in the nucleus. Why? In preparation for cell division. And then that DNA wraps around these histone proteins and it condenses, wraps around the histone proteins to form chromatin, and then it condenses, and what you get is the formation of a chromosome. So you got 46 of these guys floating around, and you have corresponding chromosomes between mom and dad, and we call those homologous chromosome pairs. So when you think about why DNA replication takes place, DNA replication takes place in preparation for cell division or mitosis. So DNA replication takes place in preparation for cell division or mitosis. So here we have a cell in interphase, and you have one chromosome, 2 chromosomes. Now, in order to divvy out DNA equally to each cell, these chromosomes need to duplicate themselves. These are duplicated chromosomes. You then take those duplicated chromosomes and you divvy them out equally into the 2 daughter cells. That is the process of mitosis, right? And eventually, when those cells separate from one another, that's cytokinesis. So when we look down here, this is kind of a way to walk through thinking about it. So this is going to reset in just a moment. You have prophase, where the chromatin condenses, so you can actually physically see the chromosomes and the nucleus starts to disappear. Prometaphase, when your centrioles, right, migrate to opposite poles of the cells and the mitotic spindle forms. You attach here to the centromeres. Then you have metaphase, when all of the chromosomes line up. Anaphase, where the mitotic spindle pulls them to opposite sides of the cell. Telophase, where the nucleus reforms around each set of chromosomes. And then cytokinesis, where the cell divides. That's mitosis. Why does DNA replication occur? It occurs in preparation for mitosis, or in preparation for cell division. So DNA replication is when DNA is used to make more DNA. Happens in the nucleus, happens in preparation for mitosis. Now, in order to understand DNA replication, it's kind of important to just review the basic structure of DNA. So DNA is a right-handed double helix, right? It consists of, it's what's called a polynucleotide, so it consists of these little building blocks called nucleotides. So here's a nucleotide, here's a nucleotide, here's a nucleotide, here's a nucleotide. Now, DNA is a long, stable molecule. It's not easily degraded and it's protected in the nucleus. And one of the mechanisms to protect DNA from degradation is that it's double-stranded. But these strands run in opposite directions, right? So these strands run in opposite directions. So if we look at a DNA molecule, we have our nitrogenous base, which is like the chemical letters of the genetic alphabet, our 5-carbon sugar, and our phosphate. Now, the sugar and the phosphate don't actually encode genetic information. They're not letters within the genetic alphabet, but they form the structural backbone of DNA, right? So they're really strong and they form these covalent bonds. Now, it's important to recognize the 5' and a 3' of a DNA molecule or an RNA molecule because it's important with respect to talking about replication, transcription, and translation. The 5' of a DNA molecule essentially has a free phosphate, and the 3' of a DNA molecule has a free hydroxide. In order to link one nucleotide to another, you need that free hydroxide for dehydration synthesis to occur. So here's your 5' here's your 3'. You're going to hear me say a lot that DNA replication goes from 5 to 3. Right? Or 5' to 3'. That's because the only place that you can link a nucleotide is at this hydroxyl. You need this free hydroxyl group, right? Now remember that the DNA strands run in opposite directions, and the different strands, the template and the complementary strands, are held together by hydrogen bonds. And these hydrogen bonds form between your complementary base pairs. So here's adenine, thymine, guanine, cytosine. Right? And we've talked about all that before, but it's really important to re-emphasize what constitutes the 5' and the 3' because we're going to talk about that a lot with replication. So when you think about steps to replication, there are 3 steps, right, to replicating DNA, using DNA to make more DNA in preparation for cell division. So when you look over here at this GIF is taking place in the nucleus. And step 1 is what's called initiation. So you see step 1 of DNA replication, helicase unwinds and unzips the molecule. DNA helicase is an enzyme. So we got step 1, initiation. Which of the following would be true of initiation? Well, you have an enzyme called DNA helicase that comes here and breaks hydrogen bonds because in order to replicate DNA, you need to split those strands. So what you're looking at right here is DNA helicase breaking those hydrogen bonds, unwinding or unzipping the DNA molecule. What you form from that, so there's DNA helicase, breaking those hydrogen bonds, unzipping the DNA molecule, that's initiation, and what you form is what's called a replication fork. So we'll wait to see this one more time. DNA helicase, breaking those hydrogen bonds. This whole thing is called a replication fork. So step one, DNA helicase breaks those hydrogen bonds and splits the DNA molecule, so you have the different strands that you're going to use as essentially templates for building new molecules of DNA, right? For building the new strands of DNA. So then you get elongation. So this little key right here represents DNA helicase. And DNA helicase comes in and it splits those hydrogen bonds, which allows another enzyme to begin working. So that other enzyme that's represented right here with the paintbrushes, that's what's called DNA polymerase. So DNA polymerase is an enzyme that essentially makes those complementary strands once they're exposed. So you get the helicase that splits those strands, exposing them, and then you get the DNA polymerase, which goes in and matches those complementary base pairs so you can get two what are called daughter strands of DNA. So there's your helicase, there's your DNA polymerase. Boom, complementary base pairs, formation of the peptide or the covalent bonds. Right? Boom, boom, and then you get those corresponding complementary base pairs. So what makes that new strand of DNA, right, from the replication fork, from those pre-existing DNA strands, is an enzyme called DNA polymerase, and that's what's being represented by those paintbrushes right there. So the DNA polymerase matches the complementary base pairs, right, on the nucleotides with their counterparts on the pre-existing DNA molecule, and in that way you can make a new strand of DNA. So you split it, DNA polymerase comes by, adenine always forms complementary base pairs with thymine, thymine with adenine, guanine with cytosine, so on and so forth down the chain. This is an enzyme. This is a piece of machinery that's actually physically doing that. Now, when you look here at this continuous strand that's being built, this is what's called the leading strand. And when you look over here, we're going to look over here for just a second, right? Just a moment when this starts over again. So we have the leading strand, and then this strand up here that's being formed is called the lagging strand. And I'm going to show you one of the things that you need to know about the lagging strand. So you have multiple DNA polymerase molecules coming in, they're enzymes, and what you see is that you form these fragments. These fragments of DNA on the lagging strand are called Okazaki fragments. So before that DNA molecule is complete, you got to knit those fragments together. And what knits those fragments together is this enzyme right here called DNA ligase. And what DNA ligase does is it essentially knits those DNA fragments together. So you have initiation with DNA helicase, you have elongation with DNA polymerase, and on the lagging strand, DNA ligase. And when you look at this, eventually you're going to get to a point of termination. So I kind of want to walk through what's happening here. So you have your replication fork. This is DNA helicase right here. It's splitting those DNA molecules. It's splitting the two strands in the DNA molecule. It's breaking those hydrogen bonds. Then you have a little enzyme called DNA primase. It puts in an RNA primer so the DNA knows where to start replication from. That DNA polymerase enzyme knows where to start replication from. Comes in, it fills in those complementary base pairs, and then we get DNA ligase on the lagging strand, knitting those complementary base pairs together, and you see it going on up here with DNA polymerase making the new strand of DNA. So the enzyme that actually physically makes the new strand of DNA is called DNA polymerase. When we look down here, when two replication forks come together, that's an indicator to the DNA polymerase for termination. So you see this replication fork, they meet with one another. That's what tells the DNA polymerase enzyme, hey, termination is good, we can go ahead and detach, and we can go out and we can wait for the next round of DNA replication to occur. So when you think about DNA replication, there are three phases to it. There's initiation. The enzyme I want you to be familiar with initiation is DNA helicase and topoisomerase. DNA helicase essentially splits those hydrogen bonds. Topoisomerase stabilizes the structure, right? It essentially unwinds the DNA so the helicase can split it. So this topoisomerase is unwinding that DNA, and then the helicase is actually splitting the strands by breaking the hydrogen bonds among complementary base pairs. Right? So you have the helicase, the topoisomerase. Now, this is your replication fork. So you have a strand of DNA and a strand of DNA. This strand of DNA, if you notice, this is the 5, so you kind of twist it over here. And the only way DNA replication can occur is from 5 to 3. So when you look at this strand up top, this is what's called the lagging strand. The lagging strand is made in fragments called Okazaki fragments. So this lagging strand is made in fragments called Okazaki fragments. And when you look at the direction that the lagging strand is being made, the lagging strand is being made away from the replication fork. So it's actually being synthesized away from the replication fork. You can see the DNA polymerase right there. And if you look at it, it's happening in a 5 to 3 prime direction. So this is your 5 prime with the free phosphate. This is your 3 prime with the free hydroxyl. You can only tack on at the end of the 3. So it's always 5 to 3 when you're talking about the polymerization of polynucleotides like DNA or RNA. So you have the 5 prime to 3 prime. There's your DNA polymerase actually making that new strand of DNA. And then on the lagging strand, these are your fragments, and this DNA ligase is coming together and knitting together those fragments, forming that sugar phosphate or completing that sugar phosphate backbone. Over here with the leading strand DNA polymerase always synthesizes, right, that strand in one chunk, meaning that there isn't fragments. And the leading strand is always going to be moving toward the replication fork. So this DNA polymerase right here, DNA polymerase, is synthesizing this leading strand. It's going from a 5-3 direction, 5 prime to 3 prime, because you can only tack on to that free hydroxyl, and it's moving toward the replication fork. When this hits this, meaning when this DNA polymerase hits the replication fork, that's going to be the point of termination, the same thing in the opposite direction. So you have initiation, helicase splits the 2 strands of DNA. Elongation, DNA polymerase essentially fills in the complementary base pairs of those 2 strands. And then termination, when those 2 strands, those 2 what are called daughter strands, eventually hit another replication fork, that's a point at which the DNA polymerase will disengage and the process will be over. Now, when you think about replication, where does replication occur? In the nucleus, right, of a cell that's an interphase. So a cell just being a cell, but it occurs in the nucleus. Why does it occur? Because the cell is getting prepared to divide. Right? So DNA replication occurs in preparation for division. It happens in the cell nucleus, happens in preparation for cell division. What are the 3 phases? Initiation, elongation, termination. One of the things you need to realize is that you are knitting nucleotides together. And one of the things I want you to understand is that these nucleotides are all linked to phosphate groups, and we've already talked about phosphate groups being highly energetic. In order to form a covalent bond between one nucleotide and another, energy needs to be absorbed. That energy comes from the nucleotide itself. So there's adenosine triphosphate, guanosine triphosphate, thymine triphosphate. This is often omitted, and it's not a major point about replication on your exam, but it is an energetic process. It does require energy, and I think that's important to realize whenever you're talking about knitting covalent bonds together. There's always going to be an energetic input. So, DNA replication happens in the nucleus during the S phase of interphase, while the cell's just being a cell. That cell is getting prepared for cellular division, and that's why DNA replication occurs. What is it? It's when DNA is used to make more DNA. So you have a parent strand of DNA, it's used to make 2 daughter strands of DNA with initiation, elongation, and termination that we've just talked about. Gene expression is different. Gene expression happens exclusively in interphase, and it's how the cell coordinates its activity during interphase, meaning that it's how the cell ultimately is a cell. It's how the cell coordinates its internal activity, right? How is it actually functioning? It's when a cell is being a cell and it needs to make little proteins, right? These little pieces of machinery to carry out all the functions needed by that cell or necessary for that cell to survive. So you have DNA, a functional unit of DNA. So a functional unit of DNA that has specific information, right, that contains information is called a gene. So you have these 46 really long strands of DNA. On each one of those strands of DNA, you have many, many genes. And through the process of transcription, these genes, or this DNA, is used to make RNA. This RNA, through the process of translation, is then used to make proteins. These proteins go out and they serve a function. They're little pieces of machinery that have a specific functional role. So this particular phenomenon you're looking at, this gene expression, whenever DNA or a gene is being used to make a protein, so whenever DNA or a gene, which is a functional segment of DNA, is used to make a protein, we say that that gene is being expressed. And that's what gene expression means. We're talking about the use of DNA to make proteins. Now, how does that process occur? How does DNA ultimately turn into a protein? And that's when we're talking about transcription and translation and linking ideas together. So DNA replication, right? DNA replication that we talked about before gene expression. DNA replication happens in the nucleus, and it's when DNA is used to make more DNA in preparation for cell division or for mitosis. Gene expression, right, is when a cell is just being a cell and it needs to make proteins to allow it to function properly. Now, gene expression occurs so similarly in all organisms that we refer to it as being the central dogma of biological sciences. Gene expression is very similar in deep sea bacteria to, mold, to humans, to blue whales. All of our cells essentially work the same way across the board. So when you're looking at this diagram over here and it says the central dogma is just that gene expression occurs essentially the same with minor variations in all organisms, meaning all organisms have DNA. They copy that DNA to form RNA, and that RNA is then used to form polypeptides/proteins that go out and serve a specific functional role. So similar is this process. We can take genes from humans, splice them into bacteria, and turn little bacteria into little protein-producing factories. So when you're thinking about transcription and translation, transcription is the process in which we use DNA to make RNA. So what you're looking at here, this double-stranded molecule is DNA, and remember a functional segment of DNA is called a gene, right? So you have these genes in your DNA, these functional segments of DNA that code for proteins, and through the process of transcription, DNA is used to make what's called RNA. And in this particular image, you're looking at an image called, or a type of RNA called messenger RNA. Now, transcribing something means to copy it. So if I asked you to transcribe this lecture, you'd just be copying what I'm saying. Well, transcription, which takes place in the nucleus, is when DNA is used to make RNA, but that RNA is really just a copy of that DNA. That RNA then wiggles out of the nucleus through those nuclear pores where it encounters a ribosome. The large and small ribosomal subunits attach, and the ribosome reads that RNA. So it actually physically reads that RNA in the process of translation, and the information in that RNA is used to make a polypeptide or a protein. This protein then goes out and serves a specific function. So the central dogma is essentially all organisms work like this at a fundamental level, at a cellular level. DNA, and specifically genes, are transcribed through the process of transcription, meaning that they're copied, right, to form RNA. So transcription takes place in the nucleus of eukaryotic organisms, and it's the process in which DNA is used to make RNA. RNA then wiggles out of the nucleus, out of those nuclear pores, where it binds to or encounters a ribosome. That ribosome then uses that RNA, right? The information within that RNA to make a polypeptide or a protein. That protein then goes and serves a specific function. Now, that protein's function could be wide-ranging, and we're going to talk about that kind of at the end, but we're going to go through these steps one by one. And we're going to start by focusing on transcription. So, in general, transcription is the process that takes place in the nucleus of a cell, and transcription is essentially copying DNA to make RNA. So, transcription is the process in which DNA is used to make RNA, right? And it happens in the cell's nucleus in eukaryotic organisms. Now, if we were to look at transcription up close, so now we're looking at a functional segment of DNA, we're looking at a gene, and this gene is being transcribed, meaning that this gene is being copied and it's being used to make a much shorter molecule of RNA. Right? DNA molecules are really long, stable, they house all your genetic information. You don't ever want those breaking down. You got to conserve that genetic information from one generation to the next. So one of the reasons DNA is double helix or double stranded is to prevent it from breaking down, because you have to conserve genetic information from one generation to the next generation. Within eukaryotic organisms, the reason it's found in the nucleus is to further protect it. Now, another reason that we use this intermediate step, we first undergo transcription and then translation, is you don't want all of your DNA being read at the same time. So you don't want all the DNA in a cell being read at the same time and every possible variation of a protein being made all the time. You want to make proteins when you need to make proteins. And what breaking this into these two steps does is it allows you to read and ultimately use different segments of DNA as the cell needs them. So in the process of transcription, you have this enzyme called RNA polymerase. And RNA polymerase essentially pries open the strands of DNA, and what it does is it matches these complementary base pairs on the RNA molecule to the base pairs on the coding or the template strand of DNA. These are called nucleotide triphosphates, just to pay homage to the fact that they carry energy, but think of them as being nucleotides. Sugar phosphate, nitrogenous base pair. The nitrogenous base pair is what carries information, so we often just give the sugar and the phosphate the ribbon look, and then the nitrogenous base pair is where the meat and potatoes is. That's where the information is being carried. Those are the chemical letters of the genetic alphabet. So in transcription, DNA is used to make RNA. And it always occurs from 5' to 3'. So 5' free phosphate to 3' free hydroxide, or hydroxyl, pardon me, right? Because you can only knit on that hydroxyl. Dehydration synthesis can only happen at that free hydroxyl, meaning you can only form covalent bonds between the sugar and the phosphate at that hydroxyl group. So transcription is the process by which DNA is used to make RNA. There are two types of RNA that we care about. So we've already seen that first type of RNA. It's called mRNA. That stands for messenger RNA. Messenger RNA is going to take the information from DNA, right? So it's essentially a copy of DNA. It's going to wiggle out of the nucleus, and it's going to bind to a ribosome where the process of translation is going to occur, and that coded information is going to be used to make a protein. So you have messenger RNA, and then you have another type of RNA called tRNA or transfer RNA. The job of tRNA is to carry amino acids from the environment to the ribosome, which allows for the linking together of amino acids. And if you look at the way that the tRNA can recognize the portion of DNA that it needs to bind to, you're talking about, mRNA that it needs to bind to is you're talking about complementary base pairing. So here we have cytosine with guanine because it's RNA. We're using uracil, which forms complementary base pairs with adenine, cytosine with guanine. A triplet set of base pairs on mRNA is called a codon. On tRNA is called an anticodon. And notice that they correlate with one another. Now we're going to come back and revisit that in just a minute. Now, transcription factors. You have a couple of different types of transcription factors. You have what are called activators and repressors. Transcription factors are essentially proteins that bind to specific segments of DNA, and they either turn genes on if they're activators or turn genes off if they're repressors. So not all your genes and all your cells are turned on or turned off at the same time, right? You turn on genes in accordance with the needs of the cells. So for example, if you need to start breaking down glycogen so you can release the breakdown product of glycogen, which is glucose, into the blood, you're going to upregulate or activate the gene, or the cell's going to activate the gene necessary to do that. So genes are being turned on and off all the time by what are called transcription factors, and there are two types of transcription factors. There's activators, which turn genes on, and there's repressors, which turn genes off. So when you think about transcription in eukaryotic cells, and we're not going to get into polycistronic or monocistronic, but transcription is taking place within the nucleus. Transcription is the process by which DNA is used to make RNA. And notice that you have this pre-mRNA here, and it gets cut down a little bit to form mRNA. Now, we don't talk much about alternate splicing in this class, but it is an important process. One gene can essentially make tons of different mRNAs and tons of different proteins. It's just an impressive little feature. So don't focus on that for the moment. Transcription, which takes place in the nucleus, is the process by which DNA is used to make RNA. And we're going to focus on mRNA. Messenger RNA, which is essentially a copy of DNA, right? And remember, a gene is a functional segment of DNA, then wiggles out of the nucleus where it encounters a ribosome or binds to a ribosome. And that ribosome uses the information in that mRNA in order to make a protein. When a ribosome uses mRNA and the information within mRNA to make a protein, to actually physically link together amino acids into this functional protein that's going to go out and do something, right, in the cell or in your body. That protein could be a hormone, it could be an antibody to fight COVID-19, whatever the hell that protein is doing, right? The process by which ribosomes use mRNA to make proteins is called translation. So at the end of the day, genes are information for making RNA and proteins. And then those proteins go out and do something. Maybe they're transporters that move glucose molecules into cells. Maybe they're contractile proteins in your skeletal muscle. So the translation is taking that RNA and using the information in that RNA to make a protein. Let's look at how transcription and translation occur. So with transcription, you're using what's called the coding strand of DNA to make a molecule of mRNA. So when you're looking at transcription here, and we're going to wait for this little GIF to kind of start over in just a moment, transcription is using DNA to make mRNA. It happens in the nucleus. So you use that DNA, you use that coding strand of DNA to make mRNA. That happens in the nucleus. The mRNA then wiggles out of the nucleus, and what you get is what's called translation. Translation is when this ribosome, here's your large and small ribosomal subunits, binds to and uses the information in the mRNA to pin amino acids together via covalent bonds called peptide bonds. And when multiple amino acids are pinned together via peptide bonds and they fold into a shape, what you get is a functional protein. So transcription, let's look at transcription. This hand represents RNA polymerase. Here's your coding strand of DNA, 5' to 3', right? Free phosphate? No. So we're going here and we're going through this. that RNA polymerase is essentially matching up those complementary base pairs. So it's matching up those complementary base pairs and it's building this strand of mRNA. That's transcription. This mRNA then moves into the cytoplasm where the large and small ribosomal subunits bind to it, and they use the information in that mRNA to build a protein. What you're seeing here are the tRNAs. So those tRNAs are bringing over amino acids and corresponding to the codons you see here. Those amino acids are linking together via peptide bonds, right, to form a protein. And what will happen is that protein will fold into a specific three-dimensional shape where it serves a function. So transcription is the process by which DNA is used to make RNA. The focal point here will be mRNA. So here we have in the nucleus transcription taking place. We got RNA polymerase represented by that little hand, right? Using the information in the coding strand of DNA to make a molecule of mRNA. That mRNA then wiggles out of the nucleus, attaches the small and then the large ribosomal subunit, and these codons, right, attract anticodons, which are located on the tRNA molecules, which will eventually, what the ribosome is doing is essentially pinning those amino acids together covalently with peptide bonds to make a polypeptide. This peptide will then fold into a three-dimensional shape and it will go out and it will do something. That's transcription and translation. When a gene, we'll call this a gene right here, is actively being used in the process of transcription and translation, meaning a gene is being used to make a protein, we say that that gene is being expressed. So to look a little bit closer at transcription and translation, we need to look at the small and large ribosomal subunits. So this mRNA will attach to the small ribosomal subunit, and then the large ribosomal subunit will come together, forming a complete ribosome. Now, because this isn't 1406, I'm not going to elaborate on that too much. Now, the ribosome essentially changes its reading frame and it moves down this mRNA molecule. But what you'll see is that those tRNA molecules enter the ribosome at what's called the A, or the access point, and then the ribosome moves them down to the P, or the peptidyl site, and what happens at the peptidyl or peptide site is you get the formation of a peptide bond between those amino acids. Remember, multiple amino acids linked together by peptide bonds forms a polypeptide. Polypeptides are proteins. It folds into a three-dimensional shape, goes out and does something. So now we're going to look at this process with respect to what I would expect you to know on an exam, which is all of it. Now, replication, DNA used to make more DNA in order to prepare for cell division, happens exclusively in the nucleus. Gene expression is when you're using DNA to make proteins that the cell needs in order to maintain the basic processes of life. So this is happening predominantly during interphase, and what's going on here is you have DNA. You know this is DNA because it's a double-stranded molecule, right? You have your sugar and your phosphate backbone, which is represented by this line. And the reason that we don't represent that using like the actual chemical structure is because there's no information there. It's a structural component. The chemical letters of the genetic alphabet, what actually carries information or transmits information, are these nitrogenous base pairs, and it's the nitrogenous base pairs that we care about. Now, if I give you a problem like this, I'm going to refer to the template strand and the complementary strand. So you have your template, aka coding strand, and your complementary strand. Your template strand is what you should be carrying out transcription and translation from. You're always going to know your template strand because it's going to be 3 to 5 prime, because the production of nucleic acids, right, the synthesis of nucleic acids always has to go 5 prime to 3 prime. Always has to move in a 5 to 3 direction. So you're always going to know what the template strand is based on the 3 and the 5 prime ends, right? So the process of transcription. So here we have a long molecule of DNA. You have 46 long molecules of DNA. They're wrapped around histone proteins to form chromatin, which condenses to form chromosomes. But when you're using DNA, when the cell's just being a cell during interphase and we gotta use DNA, meaning we gotta express genes, that DNA is kind of unraveled, it's a little looser, and these functional segments of DNA are called genes. Right? So these coding segments of DNA, right? These specific coding segments are called genes, and each strand of DNA has anywhere from hundreds to thousands of genes on it. Now, if we look more closely at this gene, right, we see that there's a template strand and a complementary strand. So if I asked you to complete the process of DNA replication using the template strand above, what I'm asking you is for the complementary strand of DNA. Right? So this would be facilitated by DNA polymerase, or that process would be facilitated by DNA polymerase. Complementary DNA strand, thymine with adenine, cytosine with guanine, adenine with thymine. If I asked you for the product of transcription, transcription is when we're using DNA to make RNA, right? So the product of transcription is going to be an RNA molecule. So what we're going to do is we're going to take our template strand of DNA and we're going to do our complementary base pairing the same way. But remember that RNA does not use thymine. It uses uracil, or U, instead. So it's a single-stranded molecule. It's essentially there to make copies of DNA, right, that can be taken out of the nucleus and used by ribosomes. And instead of using thymine, it uses uracil. So adenine forms complementary base pairs with uracil, cytosine with guanine, adenine, uracil, adenine, uracil, adenine, uracil, cytosine, guanine, cytosine, adenine, uracil, guanine, cytosine, thymine, adenine. So the only letter that changes, essentially, is all the T's turn to U's, because uracil is the base pair used only or exclusively by RNA instead of thymine. So the process of transcription is when DNA, a template strand of DNA, is used to make a copy called RNA. And specifically in this case, we're talking about messenger RNA, because that's what's going to wiggle out of the nucleus and bind to the ribosome, giving the ribosome the information it needs to make a protein. The process of translation is where ribosomes use mRNA to make proteins. Now, within this mRNA, what I want you to do when you're trying to figure out the process of translation, you go, okay, in human cells, eukaryotic cells, this process is taking place in the cytoplasm. So you have this mRNA and this ribosome that binds. We can't see it right now, but I know how the ribosome's going to read it. The ribosome's going to read it in triplet base pairs called codons. So separate out those base pairs, those letters of the genetic alphabet, into triplets. Triplet, triplet, triplet. Codons are like little words in a gene. So think of a gene being like a chapter of a book and a codon being like one word in that chapter. They're small, discrete units of information. And each codon within a gene correlates to a specific amino acid. So when you get UGG, for example, that particular codon always corresponds to the amino acid tryptophan. So the ribosome is actually going to attract that tRNA, and it's going to attract that tryptophan, and that tryptophan is going to be included as one of the amino acids in this protein at that specific point. Then you get phenylalanine, glycine, serine, right? So you split it into these triplet base pairs called codons, and these codons actually code for specific amino acids. These amino acids then link together covalently. Those covalent bonds between one amino acid and another are called peptides. And remember, it's going to fold into this three-dimensional shape, this polypeptide or protein down here, and that protein is going to go and serve some kind of functional role. Now, one of the things I'm going to give you on the exam, because I'm definitely going to expect you to be able to write out the process of transcription and translation. So I'm going to ask you, using the template strand of DNA above, right, what is the product of transcription? Well, transcription is when DNA is being used to make RNA, and we'll think of it as mRNA at the moment. So we match our complementary base pairs because the product of transcription is mRNA. So we say, okay, adenine's going to form complementary base pairs with uracil, cytosine with guanine, and you go through that whole process. Now you have a single-stranded molecule that's ready to wiggle out of the nucleus to do what it's going to do. In order to figure out what amino acids that it codes for, you're going to have to split that molecule into triplets. Those triplet base pairs are called codons. So the next thing I'm going to give you is what's called a codon table. And this codon table matches these codons to the amino acid that they correlate to. So it's essentially giving you an insight into how the ribosome actually knows what amino acids to include in a protein at what specific location in that chain. So here we have UGG. You go to U, there's your first base. You go to your second base, which is G, and then UGG codes for the amino acid tryptophan. Right? So at that location, you're going to get the amino acid tryptophan. UUU, you have your first base, U. You have your second base, U. UUU is going to be phenylalanine. Right? So then you can figure out what amino acids, tryptophan, phenylalanine, are in the actual sequence, and you can kind of double check your work on that one right there. So I've given you a problem on your sheets that you're taking home with you. Now, you have transcription, which is the process by which DNA is used to make mRNA. Translation is the process in which mRNA is read by a ribosome, and that information is used to make a protein. Proteins then go out and serve a specific function. That function could be structural. That function could be mechanical, like being contractile proteins in muscles. That function could be immunological, like the antibodies in your blood. And ultimately, what determines your traits are proteins. Proteins determine your traits. So we're going to look at this little video real quick, and then we're going to talk about that.
If you could step inside one of your cells, you'd see something that looks a lot like a factory, building thousands of different molecular machines. It would have tiny assembly lines starting in the cell's nucleus and stretching out to structures called ribosomes. These assembly lines start with DNA and build RNAs and proteins. In a very real sense, you. DNA has all the blueprints for everything the cell will ever need to build. RNAs are assorted molecular machines that do all kinds of work in the cell. They carry components from place to place, ratchet up the speed of operations, switch other machines on and off, and relay coded instructions from the nucleus out to the factory floor. The other product, proteins, are strong molecular machines that do things like hold the cell together and send signals to other cells. There are two parts to each assembly line. The first builds RNAs and the second builds proteins. The process that builds RNAs is called transcription. It happens in the cell's central office, the nucleus, where DNA blueprints are stored. Let's say that a cell needs to make a transfer RNA, or tRNA, a triangle-shaped RNA which transports a building material called an amino acid. It starts at a tiny specific region of DNA with the code that makes the tRNA we need. This region is called a gene. A protein machine inside the nucleus pries apart the weak bonds that hold the two strands of DNA together. RNA building blocks swarm in and form a conga line complementary to the DNA. This RNA strand now needs to fold up. In the world of tiny cellular machines, shape determines function. RNA's 4 bases bond to each other and give the tRNA its 3D shape. And voila, our transfer RNA is ready for action. It floats out of the nucleus and picks up an amino acid. We'll come back to our tRNA in just a moment. Now let's look at the second half of the factory's assembly line, making proteins from RNA, a process called translation. Remember the scrape? This cell needs to make thrombin, a protein machine which helps blood clot and make a scab. Thrombin is just one of the tens of thousands of proteins your cells can make. Before translation begins, the cell transcribes a special type of RNA called mRNA, or messenger RNA. This mRNA carries the code for thrombin out of the nucleus and onto the cellular factory floor. Our mRNA drifts until it runs into a ribosome, a protein-making machine. The ribosome clamps down around the mRNA. tRNAs, just like the one we made earlier, drift in. The messenger RNA carries a coded message, which the tRNAs translate into amino acids, the language of proteins. This process, translation, proceeds down the mRNA, creating a chain of bonded amino acids. Just as with RNA, molecular shape determines molecular function, so these amino acids fold up into the protein's three-dimensional form. Finally, the ribosome comes to a bit of code that says, "Okay, we're done here," and releases its completed protein into the cell. So there you have it. DNA encodes genes to make RNA machines, and the ribosome translates special mRNA, which in turn makes protein machines. Unlike most factories, your cell uses just two processes to make tens of thousands of different molecular machines. It's like one factory that can build toasters, phones, cars, and even repair itself.
So one of the things that he was talking about here, and this is really important, when you look at the example I give you, notice that the ribosome starts reading at this codon AUG. If you look at AUG on this particular codon sheet, you're going to see that AUG corresponds with methionine. And methionine is always the start codon, meaning that all of these polypeptides are always going to begin at methionine. And you can have multiple methionines in a polypeptide, but it's always going to start at AUG. So think about that for an exam. If you're giving me the product of transcription, which is mRNA, and then of translation, which is a protein, if your protein, if your mRNA doesn't start with AUG and that protein, right, doesn't start with the amino acid methionine, you might have done something wrong. Then when you notice at the end on your practice sheets, you're going to get one of these codons, UAG, UAA, UAG, or UGA. Those correspond with stop codons. They don't carry an amino acid, and that's how the ribosome knows, hey, I'm done. So every polypeptide that you're going to be building for me, like when you're doing a practice activity, is always going to have a start and a stop for me. So pay attention to that on your handout. Now, why do we care about this? Because proteins code for your traits. Traits are those measurable things that you think of when you think about a person. Their hair color, whether they have a widow's peak, right? Their eye color, those things that you physically see, they're determined by proteins. So let's talk about the color of my eye. How does a gene ultimately determine the color of my eye? Well, the cells in the iris of my eye, right, have nuclei, and in those nuclei there's DNA, and I have the gene for brown eyes. Through the process of transcription, that gene, that DNA, is used to make mRNA. Then that mRNA wiggles out of the nucleus in the cells in my eye, right, in the iris of my eye, and it binds to a ribosome. That ribosome then uses that mRNA to make a protein, right? The protein in this case is an enzyme that converts an amino acid into a pigment called melanin. That pigment then deposits itself in my eye, so that enzyme converts an amino acid to a brown pigment called melanin. That brown pigment deposits itself in the iris of my eye, where it prevents the scattering of light and protects my eye from UV radiation. So the pigment in my eye is the direct consequence of gene expression, right? So your traits, your biochemical traits, your physical traits are determined by gene expression. They're determined by your DNA. But we don't express our genes equally, and in fact, we have a tremendous amount of influence over the way that genes are expressed. So for example, here you're looking at somebody on steroid therapy who's undergoing gender reassignment. And what they're doing is they're taking large amounts of steroid hormones like testosterone, and the way that steroid hormones work is they influence gene expression. It takes a while to influence gene expression. So those steroids act like transcription factors. They turn genes on and they turn genes off. Now, it takes a lot of time, but if you look at this, you eventually see the transition from predominantly feminine to predominantly masculine characteristics over a period of time, right? That's because the way that those genes are being read is different. So hair growth, the pull on the vocal cords, etc., changes. Muscle deposition, it all changes. So you can influence gene expression. For example, over here you have genetically identical twins. But one identical twin is overweight and the other is at a healthy body mass index. The reason that you see this is because one chose lifestyles that influence certain genes, like the gene to take sugars and use those sugars to make fats to store energy, just as a consequence of having so much sugar in the system, whereas the other didn't. So your lifestyle factors can influence the way that genes are expressed. So genes are interacting with your environment all the time. Just because you have a set of genetic characteristics doesn't mean that they're necessarily going to manifest, right? Genes are it's this complex interplay between your environment and your cells that's taking place all the time. Now, when you think about proteins, proteins are little machines, man. They're molecular machines. And they go around and they do stuff like facilitate muscle contraction or catalyze reaction or function like chemical signaling molecules, or they're antibodies that mediate immune responses, or they're channels or transporters. So when you talk about cell biology, when you get into AMP1, you're going to talk a lot about proteins. Like this here is a little motor protein. It's physically moving stuff around in the cell. This choo-choo track is a protein. Those things embedded in the membrane are proteins. They do everything. So you're going to talk a lot about, processes from the perspective of a protein. Even when you're looking at tissue histology, right, which is something else you're going to be doing in AMP. Here we have areolar connective tissue. There are collagen fibers, the thick kind of, so you have these cells and then these collagen fibers and these elastic fibers. Those fibers are proteins. Here's your skeletal muscle, right, that takes on this striped appearance under a microscope. All of those kind of, the reason it takes on a striped appearance has to do with the organization of proteins in the cell. So most of what you focus on in like AMP is at the level of proteins, and then being able to expand that and go, okay, this is happening at the molecular level. How does this influence the entire cell? Drug targets, when you get into pharmacology, right? 85% of drug targets are proteins. So there are lipid and nucleic acid and carbohydrate drug targets, but most drug targets are proteins. So for example, when you're reading about diabetes medications and it says the mechanism of action of this medication is it inhibits the sodium glucose cotransporter, you go, okay, the sodium glucose cotransporter is a protein that allows us to reabsorb glucose in the kidneys. If you inhibit that, you're not going to be able to reabsorb glucose or sugar from your urine, and you're just going to urinate that glucose out, which is going to drop your blood glucose levels. It's connecting things. Protein, protein, protein. So you talk about them all the time in anatomy and physiology and pharmacology, and most of what you focus on in A&P is mechanism, and mechanism almost always ties to protein. So whenever you hear the term protein, go, oh yeah, we have a gene, and through the process of transcription, that's mRNA, through the process of translation, that makes a protein, then that protein goes out and does something. It's also critical to our understanding of genetic diseases. So what you're looking at here is a chromosome. Remember, inside the nucleus of every one of your cells, you have 46 chromosomes. Chromosomes are made from long strands of DNA wrapped around histone proteins. So you have 46 long strands of DNA. Now, in those 46 long strands of DNA, you have around 3.2 billion nucleotides, meaning 3.2 billion letters in the genetic alphabet. So every time replication happens, right, that cell has to copy 3.2 billion letters. It would be like copying 700,000 textbooks. Cells make mistakes. Even though there are mechanisms to proofread and prevent mistakes from being made, cells make mistakes all the time. So you could get a situation in which one letter, right, in your coding strand of DNA is swapped with another. And what that's going to do is it's going to have downstream consequences. So we refer to that when DNA is altered via UV radiation or chemicals or whatever it is, right? We refer to that alteration of DNA as being a mutation. Mutations in adult cells, which is where most mutations occur, right, are just changes of DNA in adult cells. Mutation of gamete cells or germ cells are mutations that occur in sperm or egg cells, and they're going to be passed on to the next generation. So you have different types of mutations. For example, you have silent mutations. Silent mutations are mutations, but because there's a bit of redundancy in the genetic code, we have this third, what's called wobble base, not again a focus of my class, right? Silent mutations are where the DNA code, the genetic code has been changed, but the information it codes for, meaning the amino acid that's going to be in the protein, doesn't, and therefore it's going to have no functional consequence. Those are called silent mutations. Nonsense mutations are when you get a change, for example, in like the first base pair of a codon that produces a premature stop codon that will almost always be catastrophic to the cell. Missense mutations are mutations in which one base pair changes. So here we have TTC, here we have TCC, and that one base pair change produces a situation in which a different amino acid is coded for. And that's called a missense mutation. Ignore conservative and non-conservative. Right? So we have all these different mutations that can happen in your DNA. And when you think about it, I want you to think back to this idea of the central dogma. So DNA, through the process of transcription, is used to make RNA. RNA is used to make proteins. Proteins interact with lots of other proteins in what we call metabolic pathways to produce observable traits like height, hair color, eye color, glucose, measurable traits like glucose metabolism, etc. Right? Bottom line here is if you change the DNA, you change the RNA. If you change the RNA, you change the protein. If you change the protein, you change the protein's function. If you change the protein's function, it's going to have some kind of consequence on the organism. So why do we care about this? Wild type just means the most common form of a gene. So we're looking at what's called the PAX6 gene, which codes for the PAX6 protein. In the normal gene, or the most common form of the gene, think of it as being kind of like the healthy gene. The PAX6 gene codes for the PAX6 protein. The PAX6 protein guides the development of the eye. It does so in humans. It's what's called a conserved trait in mice and zebrafish and Drosophila flies, right? So one, that kind of reeks of origins, but two, right, that's a conserved trait. You see that gene across the board and it guides the development of the eye. Now, why do we care about genes? Well, if you, and specifically mutations of genes, if you change a letter in the DNA alphabet, you change the DNA, you change the mRNA, you change the mRNA, you change the protein. If you change the protein, then you can either interfere with its function or completely ruin its function. Like in this case, here's a mutated PAX6 gene. In the human, we get the absent cornea, iris absent, retinal degeneration. This is congenital blindness. This is blindness from birth. So when you screw up genes, you screw up proteins. When you screw up proteins, you screw up entire systems. So now you have an organism that's not going to be able to see and won't have the same adaptive ability in its environment. Same thing in mice, zebrafish, Drosophila flies. That gene gets mutated, they can't form eyes. We care about mutations because it impacts our organ systems. And there are thousands of genetic diseases. Thousands. Wherever you have a gene, you have a corresponding genetic disease, and they range from mildly annoying to fatal intrauterine fatality, meaning spontaneous miscarriage, right? So we have single gene disorders, which are usually point mutations in which one letter out of 3.2 billion, I'll emphasize that again, one letter out of 3.2 billion gets swapped. And that ultimately, you influence the gene, the protein, right? That protein's function, and that can have catastrophic consequences. You have genetic diseases in which chromosomes, right, don't undergo a process called separation, and you get what are called disjunction or non-disjunction events. So you have chromosomal abnormalities or disorders, and then you have your single gene disorders, your chromosomal disorders, which can be broken into sex-linked and non-sex linked. Copy. So let's see single gene disorders. Wild type hemoglobin. Here's your DNA. Hemoglobin is a protein you find in red blood cells that carries oxygen around. So at this segment of the gene, you have CTT. Your mRNA is GAA. Your amino acid is glutamine, right? The protein that's produced when you throw that in there is hemoglobin. When hemoglobin is normal, right, the red blood cell takes on this biconcave shape that can fit through capillaries effectively in the cardiovascular system and deliver oxygen effectively to the tissues in your body. With sickle cell anemia, you have the mutant hemoglobin, meaning a gene has been mutated, meaning you've swapped a letter. In this case, out of thousands of letters that code for this protein, you swap a T with an A. Now you don't have this amino acid, you have this amino acid. When you throw this amino acid in on that position, it makes hemoglobin super sticky, and it sticks together and it forms these long rods. When all of the proteins in a red blood cell are sticking together forming these long rods, it changes the shape of the red blood cell to kind of look like a sickle that's used to harvest wheat, right? As you do these days. You just use your sickle to harvest your wheat or to attack your neighbor. I suggest attacking neighbors with sickles. So you have your sickle. These sickled cells are really hard and they don't fit through capillaries very well. So in sickle cell anemia, you don't deliver oxygen to tissues very effectively. You suffer these things called crisis. And what happens is the entire organism is now affected. So if you can't deliver oxygen to tissues effectively, that's going to affect your brain, your liver, your GI tract, etc. It all starts with a change at this tiny little genetic level, and it kind of balloons out. Now, chromosomal disorders. Chromosomal disorders are when whole chromosomes get essentially moved in the wrong direction, right? They don't split normally during either meiosis or mitosis. And in chromosomal abnormalities that often result from what are called non-disjunction events, humans should have 46 chromosomes. So the image you're looking at here is what's called a karyotype. What you're looking at are chromosomes organized from largest to smallest, right? Arbitrarily, just your largest chromosomes to your smallest chromosomes. And the final two pairs of chromosomes are what are called sex chromosomes. So you have chromosome from mom, chromosome from dad. That's a homologous chromosome pair. 1, 2 chromosomes, 3, 4. And then you organize them into these pairs that have corresponding genes on them. Now your first, 22 pairs of chromosomes, your first 44 chromosomes or 22 pairs, one from mom, one from dad, are called autosomes. They have nothing to do with the process of sexual differentiation. And that's a bit of a misnomer, but for right now it's good enough. The final pair of chromosomes, are called sex chromosomes. In the overwhelming majority of people, you have either XY or XX, but in this individual you see XXY. Well, that's actually a condition called Klinefelter syndrome, and it's similar. It's a congenital, very mild form of hermaphrodism, where you carry traits from both sexes because you have your cells confused about what genes to express and not express. Then you get oncogenes. Oncogenes are genes that if they become mutated, it will lead to cancer. So one of the most important oncogenes that we have is what's called the p53 gene. P53 gene codes for the p53 protein. The p53 protein says, hey, cells, stop dividing. It's like the angel protein of the cell. It prevents, damage to DNA. It does a lot of things. Half of the cancers that you can think of are a consequence of mutated p53 genes. Now, I'm going to show you a video on CRISPR now that you have a little bit of a background in genetics, and I know the video isn't perfect, but it's good enough.